Dear teachers and students, it is a great honor to be here to introduce one of our recent studies, a universal oral microbiome-based signature for periodontitis. Periodontitis is a common oral health concern around the world, and according to previous studies, it is known that patients with periodontitis have a greatly increased risk of systemic diseases. Early studies developed machine learning models of periodontitis using 16S sequencing based on the genus-level oral microbiome, but were influenced by low taxonomic resolution, limited data sets, and lack of global universal population, making the model universality and generalization ability low. So this study collected 8 metagenomic cohorts across 5 countries and 3 continents for marker identification, modeling analysis, and prediction. We identified 54 biomarkers with consistent trends in 3 and more cohorts, including 42 disease enrichment markers and 12 health enrichment markers, respectively. The identified disease enrichment markers included known periodontitis-associated species and many unknown new species consistent across cohorts, and we also found that some of these oral biomarkers were also enriched in the gut of patients with intestinal disease. To test whether taxonomic relative abundance data for the oral microbiome could be used to distinguish cases and controls for each cohort, we built a random forest classifier using all species features and obtained an internal AUC of 0.86 and an external AUC of 0.79. Lodo modeling analysis improved the external AUC to 0.85 and was externally validated in 2 disease cohorts with true positive rates of 89% and 72%, respectively. The slightly lower true positivity rate in the latter cohort may be attributed to disease severity. Group of disease samples according to disease severity. The true positive rate was nearly 80% for systemic aggressive periodontitis and nearly 60%, with results showing high precision and good sensitivity. Next, we explored the interaction between the above biomarker species using the correlation network analysis. A total of 299 interaction relationships were identified between the 54 biomarkers using Spearman correlation coefficient absolute values greater than 0.5 and p-values less than 0.05. And observed important correlations of the new species in the network. Figure C and D box plots show that health enrichment biomarkers showed stronger correlation and higher synchrony, indicating healthy enriched species have better adaptation in the oral environment compared to disease biomarkers. This study identified biomarkers consistent across cohorts, including 54 species markers and 26 metabolic pathway markers. The identified species biomarkers included both known species and some consistent new species. A random forest classifier based on oral metagenomics data was created to distinguish periodontitis disease groups and control groups, obtaining an internal AUC of 0.86 and an external AUC of 0.79. The Lodo modeling analysis improved the external AUC to 0.85. Found between health or disease group main biomarkers interact for positive correlation. Emphasizes the potential synergy between species or common growth requirements, highlights the necessity of the overall method to improve the oral physiological conditions, is conducive to the growth of health biomarkers at the same time, inhibit the growth of disease biomarkers.